Okay, thank you everybody. Thank you very much for joining us to this Sickle Cell Club meeting, and also a meeting of College of the Mainland students. We are very honored to have Dr. Tim Townes join us. He's from the University of Alabama, Birmingham. And I got to know Dr. Townes through his research, through reading his papers. And I saw the amount of work he had done in sickle cell and the love of that area of sickle cell, the research area. So I decided to kind of talk to him and invite him to come and talk to you students about the big things happening, because for some time there wasn't much happening into the sickle cell area, but all of a sudden so many things are happening. So I decided to be a good thing to invite somebody who is at the top end of the research to kind of help us to see where are we going with sickle cell. They're even talking about a cure of sickle cell disease. So I don't have much to say. We are so honored to have him to come and talk to us. That is a very good thing. Now, for students, if you have a question, hold on to the question and type the questions in the dialog box, and then the questions can be asked at the end. So let me stop there and then give it to Dr. Townes.
Okay, thank you so much, James. It's an honor to be invited, and I look forward to giving this talk. A lot is going on in sickle cell disease therapy now, and I'm going to talk primarily about our work. I will compare it to the work of others, and then you can ask more questions about the work of others after I finish. But this is primarily data from our lab, and I'm going to meet with the Food and Drug Administration on March the 2nd, which is not far from now, to ask their permission to move towards doing a clinical trial in patients at UAB with this technology that I'm going to discuss. So we're very excited about that meeting. The clinical trials would probably be a year away, but that is because of the Food and Drug Administration is very conservative, and they want to make absolutely sure that this is a safe treatment as well as an effective treatment. Okay, so there are a lot of different strategies for correction of sickle cell disease. And one strategy that has been used already in clinical trials is what is called gene addition therapy, where the sickle mutation is not corrected, but a new beta globin gene is delivered to the patient's blood stem cells. And the way that the new gene is delivered is in a lentiviral vector. And of course, that lentivirus is the virus that causes AIDS, but no one can get AIDS from this particular treatment because the AIDS virus has been gutted, but basic sequences at the ends called the long terminal repeats and a few other sequences are included. And then a beta globin gene is put into that vector and then packaged into virus, and that virus can be used to infect blood hematopoietic stem cells, and that is done ex vivo outside the body, and then those cells, engineered cells, can be put back in by transfusion. And that has been done, and several patients have been cured. And so that's a very exciting result. There are strengths and weaknesses to every approach. The strength of that approach is it has been done and it has cured several patients. The weakness, and there's a weakness in every every drug that's used for treatment of any disease, any medical, any surgery. We live in a risk-averse society, but the fact is there are risks to everything we do. And So, but the Food and Drug Administration makes sure that the risk is very, very minimal in order to actually do a clinical trial and treat patients. So the one weakness of the lentiviral gene addition approach is that the gene is inserted at random in the genome. So the sickle mutation is not corrected. This new gene is inserted at random and is designed to function, and it functions well so that patients make normal hemoglobin and their cells don't sickle. However, the about 5 copies of the gene need to be inserted per cell in order to get a high level of globin expression. And so that means that in every cell that is infected or where it's transduced outside the body, that adds, if there were 100 million cells that are treated with the virus, and each cell gets 5 copies, that means there are 500 million random copies inserted into the genome. So far, that has not been caused problems, but there are potential problems with that because that's random integration. So that genes that are supposed to function may not function if the virus inserts into a gene and blocks its function, or the gene could activate an oncogene that could cause leukemia. That it happens very rarely, but can happen. And so those are the strengths and weaknesses of gene addition therapy. There's one other clinical trial that has been done already and actually published in the New England Journal of Medicine a few months ago. And in those experiments, 2 groups actually upregulated fetal hemoglobin. And as you know, we all make at birth about 90% fetal hemoglobin, and then over the first year, our hemoglobin switches from fetal to adult hemoglobin. And even if an individual has sickle cell disease, that is, if both copies of their beta globin gene have the sickle mutation, that does not cause any pathology in a newborn child because they're making 90% fetal hemoglobin. But as those levels shift and fetal hemoglobin or hemoglobin in F falls and hemoglobin S rises, then at about 12 months after birth, then those cells can begin to sickle. So the approach of these groups is to either keep fetal hemoglobin on or in adults reactivate fetal hemoglobin. And so they have identified sequences that are responsible for this switch from fetal to adult. That is, they're responsible for turning off the fetal globin gene progressively in the first year. And they've actually deleted those sequences, those repressors of fetal hemoglobin, and have successfully kept fetal or reactivated fetal hemoglobin in adult patients. And some patients have been cured, some sickle cell patients and some beta thalassemia patients. So the strength is that it's already been tested and there are patients that are well. The weakness is that fetal hemoglobin has a higher oxygen affinity than normal adult hemoglobin. So, and that has a purpose, which you already know, I'm sure, and that is that when a baby is in utero, the baby is not breathing, of course, mom's breathing, and the oxygen that she's breathing is being transmitted to the baby's red blood cells in the placenta where the vessels are right next to each other. And so fetal hemoglobin has to pull oxygen off of normal hemoglobin in order to deliver oxygen to the baby. So fetal hemoglobin, or hemoglobin F, has a higher oxygen affinity than normal hemoglobin. Well, the disadvantage of that in an adult is that the fetal hemoglobin hangs onto its oxygen longer and doesn't deliver it to the tissues quite as well. And therefore, no one who has high fetal hemoglobin as an adult would be a marathon runner, and actually may not be able to participate in strenuous exercise, because the oxygen is not delivered quite as well. That's perfectly fine for a fetus in utero, because they're stationary and they're not moving around that much. But for an adult, that can cause some limitations. However, it's been done successfully. What we want to do is actually correct the mutation that causes sickle cell disease. And okay, now I just tried to change the slide for the first time, and for some reason it didn't change. There we go. Great. Okay, so as you all know, the sickle cell disease was the first disease to be understood at the molecular level. It is the first disease in which the mutation that causes this inherited disease is known. And that mutation is this A to T change at codon 6 of the beta globin gene. Can you see my cursor on the slide? James, I don't know if you can see that or not. Okay. Actually, I can't see or hear anyone, so I just want to make sure that
we cannot see the mouse on the screen.
You can't see the mouse on the screen. Okay, so
No.
I'll just talk. So the disease is caused by this single base change that changes the codon at position 6 of the beta globin gene, and it changes it from GAG to GTG. And that, of course, GAG encodes glutamic acid, and GTG encodes valine. So it's an A to T change, and that valine substitution produces a hydrophobic projection on the surface of a tetramer. So if my fist is a tetramer, this valine change, if this is the hydrophobic pocket or if that's where the glutamic acid usually is, there's no hydrophobic pocket, but now there's a valine there, and that forms a hydrophobic pocket. And when that happens, I'm so sorry, the valine is not there. The valine is a projection on the surface and interacts with a hydrophobic pocket that is present on another tetramer. And that stimulates the polymerization of tens of thousands of molecules of hemoglobin to cause hemoglobin polymers, or even fibers. So those polymers interact with each other to form a 14-stranded structure that looks like a fiber optic cable if the cell is cut and an electron micrograph is done. And so that causes the cells that are normally pliable and squeeze through small vessels to deliver oxygen and then to pick up carbon dioxide to deliver back to the lungs. Now the cells are rigid and non-deformable, and some sickle. And that has two effects. One is that can block and occlude small vessels, and those occlusions, if they occur in the brain, can cause strokes. If they occur in the medulla of the kidney, which is a low oxygen tension tissue, they can destroy urine concentrating ability, and occlusions of small vessels out in peripheral limbs can severely inhibit wound healing, and that can result in amputations. Those occlusions can happen in small vessels in the retina and cause blindness. So it's a devastating disorder. And all of those things, or all those pathologies, are associated with pain and acute crises, pain crises. And so this next image is an MRI of a patient at UAB, a child with sickle cell disease. And you can see, and as you know, our brain is symmetrical, so the vessels on the left and the right should be the same. This patient's lying down on their back, so you're looking at the screen. Their right internal carotid artery looks beautiful, and it's that big vessel. I can't really point to it, but there should be a left internal carotid artery that's visible, that's a mirror image of that on the left side of her brain. And as you can see, it's not visible, and it's not because it's not there. The vessel is there, but it's occluded by sickled cells so that there's no blood flow through it. And this imaging technique visualizes flowing blood. And so this individual has had a occlusion of this large right internal or left internal carotid artery, and therefore it has oxygen deprivation to the left side of her brain. So she is completely paralyzed on her right side. And that only happens 1 to 2, maybe 5% of patients. But what happens in the majority of patients is that small vessels that you can see farther away, that out on the periphery, where those smaller vessels are included, and that causes cognitive impairment and learning disabilities. And more than half of sickle patients have this pathology. And so because of that, our goal is to cure the disease by correcting that one single base of a T back to an A. And our goal is to do that in newborns, in 3 to 6 month old children. And so that's illustrated here. And so our approach is to collect umbilical cord blood from newborn children. There's an advantage to collecting blood at that point because we don't have to intervene in the baby at all. The cord is cut, and while mom's delivering the placenta, our physicians that we collaborate with in the OBGYN department at UAB then collect the umbilical cord blood. They put a needle in the umbilical vein, and we can get 50 to 100 cc's of cord blood. So the cords are just going to be thrown away if we don't collect the blood from them. But we collect the blood, then we in our laboratory purify the hematopoietic stem cells, which are the cells, as you know, at the top of the pyramid in the blood system. They are producing all of the trillions of blood cells. So those are the cells that are a target of gene editing, because all red blood cells, if we can correct that T back to an A in a hematopoietic stem cell, then all the red blood cells that those cells make for the lifetime of the patient would be corrected. So the way that we do that is with CrisprCas. But we've made a modification of the CrisprCas system, and I'll tell you about that. And we can correct the sickle mutation, and then we test efficacy and safety. And then if all the FDA criteria are met for efficiency and for safety, then we propose to transfer those cells by a simple transfusion into the baby. And then all of the red blood cells made from that corrected stem cells will produce normal human hemoglobin. So in this approach, as I wrote on the bottom, the child is cured with their own cells before developing any tissue or organ damage. And that tissue and organ damage it can't be replaced. So if one has a stroke like the one I just showed, that's irreparable. And so we want to be able to cure the disease before that can happen. So in our approach was to make a biochemical complex. So I'm a biochemist and a molecular biologist, and everyone in my lab is also. And so our goal was to produce these CrisprCas reagents in our laboratory, which we have done. And and it's a 3-component complex made up of a guide RNA, and then the Cas9 enzyme, which cuts DNA, and then a single-stranded oligodeoxynucleotide, which is the correction template. So the guide RNA has the Sickle mutation in it so that it efficiently guides the Cas9 to the Sickle mutation, and then the single-stranded oligodeoxynucleotide has the normal base there, so that when the correction is done, the mutation will be correct. The gene will be corrected, and so the question is this efficient enough? And well, let me just say something about CRISPR-Cas, and I'm sure you know all about CRISPR-Cas, but I would like just to say a couple of things about it. And this is really a mechanism of immunity for bacterial systems. So this is the way bacteria protect themselves from viruses that infect them. So this is an image, an electron micrograph that's been colorized of a bacterial cell and of phage binding to it and injecting their DNA into it. So you can see the green string of DNA being injected into the cell. And of course, what happens usually is that DNA is replicated and hundreds of phage are formed inside the cell. The cell's lysed, and that's how the virus propagates itself and then infects other cells. Well, the CRISPR system was really discovered in a yogurt factory, and because yogurt, as you know, is made from fermented milk that's fermented with bacteria, and the bacteria are innocuous bacteria, and those cells ferment the milk, and that's how we make a solidified yogurt that we all eat. But in the yogurt factory, they noticed that their bacterial, that they couldn't form yogurt anymore with their standard bacterial stocks. And the reason was that phage were infecting their bacteria and killing it so that the fermentation process didn't work. So they just thawed out some new bacterial stocks and they got their yogurt production up and going again. But fortunately, they have scientists, they have biochemists and microbiologists at these companies, and they were interested, they wanted to know why did the bacteria quit fermenting? And so they got bacteria that did ferment and bacteria that didn't ferment anymore, and they sequenced their whole genome. And in the bacteria that that could ferment were resistant to these phage that were infecting the stocks that couldn't ferment. yogurt or milk. And when they looked at the sequence of the ones that could defend the bacteria from the phage, they had what they described as clustered regularly interspaced palindromic repeats, or CRISPR locus. And those sequences were transcribed into RNA. So this is the CRISPR locus, clustered regularly interspaced palindromic repeats that look just like this. And what they found was that when they sequenced this DNA, there were little bits of virus DNA in the bacterial genome. And then they discovered that those were transcribed into RNA, that RNA didn't make protein, it formed these stem loops with this attached sequence. So these are repetitive sequences that form these stem loops, and this is the viral sequence. Oh, I'm sorry, I forget, I'm pointing with my pointer. The blue and the yellow and the orange are little pieces of virus DNA that is inserted in the genome and now is transcribed into RNA. Well, it turns out that the red genes, called CRISPR-associated genes, or Cas genes, are also independently transcribed. They're transcribed into messenger RNA that is translated into protein, and that protein is depicted as those red ovals. And the Cas9 protein binds to the guide RNA, and then that the viral-like sequences in the guide RNA can guide the Cas9, which is an endonuclease that cuts DNA internally, so that if the same phage infects the cell, that CrisprCas can bind to its sequence and then digest it, which is illustrated at the bottom. So that is how bacteria gain immunity to viruses. Well, the people who ultimately worked this out demonstrated this, and this was done, this is a figure from Jennifer Doudna paper in Nature. It's a review, and this, the protein Cas9 is in blue, the guide RNA is shown in black stem loops, and then the red guide sequence. And so in our case, the guide sequence is the sickle gene RNA, and it is about the red piece there in the sickle gene is about 20 bases, and it's then attached to these stem loops. And this protein has a helicase. It binds to what's called the PAM, or the protospacer adjacent motif. In bacteria, the bacteria Streptococcus pyogenes, or this capital S lowercase p, their Cas9 binds to the sequence NGG, so N can be either any nucleotide, and those 3 bases are about 2 to 3 bases downstream from the cut site. So that's the first thing that is recognized. So the RNA, guide RNA protein complex gets into the cell, or is in the cell, and then it scans the whole genome for this guide RNA sequence so that it can form a complementary base pair to it. And you can see that when it encounters a complete target there on the left, then the enzyme Cas9 is a helicase and can unwind the DNA. And then the sequential R loop expansion, the RNA displaces the other DNA strand to form a DNA RNA hybrid. And when that's complete, that structural change in the Cas9 activates its double-stranded DNA endonuclease activity, and it cuts both strands of the DNA. And so at the bottom right, you see those nicks in both both strands, and that is, in our case, just downstream of the sickle mutation. And that happens inside a hematopoietic stem cell from the patient, which we have collected from the cord blood, purified, and then electroporated with this biochemical complex. So we electrically shock those cells in a way that just puts tiny holes in their membrane. This complex that we make goes into the cell, goes into the nucleus, scans 3 billion base pairs of DNA, both complements, so it's really 6 billion. And then it lines up with a sickle mutation, cuts it, and then one of two things can happen. And that is what's illustrated by the yellow arrows and the acronyms HDR and NHEJ. So what happens most efficiently is NHEJ. That stands for non-homologous end joining. And in that case, there are enzymes in the cell that polish the ends off and then ligate them back together. And that's important to do fairly quickly because double-strand breaks then can interact with double-strand breaks in other places of the chromosome and cause translocations. So the cell wants to get those ends back as fast as possible. But there's another way to get the ends back together, and that's by HDR, or homology-directed recombination. And that uses a small piece of DNA to actually encode as a template, encode the sequences so that it's copied into the genome. And in our case, that correction template is the single-stranded oligodeoxynucleotide. So it's about 100 bases. We synthesize that, and that has the normal letter in it, an A instead of a T. And so really, in effect, what happens is this is like a Microsoft Word document of 3 billion letters, and you have told your computer to find this sequence of letters, find it and replace it. And that's what happens. Okay, now, can we really do that? And in fact, we can. So as I said, with babies are born in our OBGYN department, we get the OBGYN physicians to collect cord blood, we purify the stem cells in the lab, and then we electroporate it with this CrisprCas. However, we've modified the Cas9 so that it slows down NHEJ and speeds up homology-directed repair. And that's very critical because NHEJ, non-homologous end joining, puts the DNA back together, but bases are deleted in that polishing process. And so that inactivates the gene. So we want to minimize that and maximize the gene correction. So that's what our modifications do. We've made 10 different modifications of the protein and tested all of them hundreds of times. And now we have the modification that we want to go forward with the to the FDA in order to be able to do a clinical trial. Okay, but before we can convince the FDA, we have to show that we do correct the mutation at an efficiency that would cure patients if we transplanted them. And so we do that in 2 ways, and that's illustrated on this slide. After we've gone through the whole process, then we, in one set of experiments, we plate those cells in what's called methyl cellulose that has all of the growth factors that are required for human hematopoietic cells to replicate. And they do then by individual cells sitting down in this jello, basically, that has all of these growth factors, and then that individual cell grows into a colony of about 100 cells, and we can pick those colonies and do DNA sequencing to see if the T has been changed to an A. All right, and so that's illustrated in these are the colonies. BFUE means burst forming unit erythroid. That's about 500, even 1,000 cells, but that came from a single cell. So this is a single cell assay, and that's very important so we can get unambiguous sequence from it. This other colony is called GEMM for granulocyte, erythroid cell, megakaryocyte, macrophage. So this is a progenitor cell that can form all these different cell types. And so we want to make sure that the mutation is corrected in all of these cell types. That's not a true hematopoietic stem cell. In this particular assay, we can't we can't quantitate correction in a true hematopoietic stem cell, but we do that with another assay that I'll tell you about in just a second. Okay, now we pick these colonies, and we'll pick 100 colonies, and then we sequence all 100 of those and ask what fraction of them are corrected. So this is kind of a busy slide, but excuse me, there's really only 3 things that are important, and those are listed in black on the right. So 56% of the cells are corrected in this assay. And by the way, we know that we only need to correct 20 to 30% of the stem cells because if those cells are transplanted and they make normal red blood cells, those red blood cells will live for 120 days. But a sickle patient's red blood cells only live 10 to 20 days. So a 20% to 30% correction in the bone marrow will end up being 70 to 80% correction out in peripheral blood because of selective survival of the corrected cells. And that's not just a guess. We know that from bone marrow transplant studies where there's a matched sibling donor. There are very few families that have matched sibling donors, but if one child is either is AA, they have both of their genes are normal, or even if they're AS, they're a carrier, they have one normal gene and one sickle gene. If those cells are transplanted into their brother or sister, and they're matched donors, then those transplants cure 95% of the patients. And if everyone had a matched sibling donor, we wouldn't have to do what I'm describing, but very few do. And but when you look at those patients that have had transplants from a matched sibling donor that are cured, their bone marrow will only have 20 to 30% corrected stem cells, but 70 to 100% of the cells out in the peripheral blood will be normal red blood cells, and they will be transfusion dependent, and they will be pain-free and be cured. So we don't have to correct 100% of the cells. We don't even have to correct 50% of the cells. So 20 to 30% is our goal. And we were very happy to see that 56% of the cells were corrected in this experiment. Okay, that's the good news. The bad news is that 40% of the cells were apparently were not corrected, and they were the result of this NHEJ that I said for repairing cuts, double-strand cuts, so that 1 or 2 bases were deleted. So they were no longer sickle genes, but they weren't viable genes. So they were basically would cause beta thalassemia. So the good news is we corrected, oh, and by the way, I'm saying this was with the wild type Cas9, with the normal Cas9 before we made our modifications. It's these results that told us we had to change and modify the Cas9. And the other problem was there was only about 25% cell survival. And we think that's because of this high number of mutations which we call indels, or insertions and deletions. So the 56% correction is great, 40% indels are bad. All right, so, and they're bad for another reason, is that they cause rearrangements in the DNA. And you know what, I'm gonna skip the slide. I'm just gonna show the schematic. This is chromosome 11, where the sickle mutation is. That's where the globin gene is. And in some cases, we saw translocations of pieces of DNA from other chromosomes into this locus. So these are chromosomal abnormalities. And here's another one that we saw in picking these colonies. So we knew then that we absolutely could not do this with the wild type Cas9, with the normal Cas9, that we had to make a modification. And our thought is that Cas9 enzymatic activity is too fast, too high for human cells, because that enzyme evolved to work in bacteria that are being infected by viruses. So if a virus infects the cell, the enzyme has to work very rapidly to chew up, to digest the phage DNA to protect the cell. But that's too fast for human cells. So we needed to slow down the activity of the enzyme, but we need to maintain stability for maximum gene correction. And then we wanted to inhibit the toxicity of electroporation with single-stranded oligonucleotides. And we wanted to enhance this single-stranded oligonucleotides association in the complex of guide RNA and Cas. All right, that's a lot to say, but I think you can see very easily. We wanted to slow down the activity of the enzyme without losing very much gene correction ability. All right, and this just shows a biochemical assay. I'll just have you look at two paints. The top left is wild type Cas9, and there are 4 lanes in this gel. This is a piece of DNA with the human sickle mutation in it, and you can see that and the 4 lanes are the highest concentrations on the left and the lowest concentration in the right of the enzyme. And you can see that in 15 minutes, 100% of the DNA is already digested. So that's very rapid digestion. Here are 2 of our modifications: modified recombinant Cas9-1, modified recombinant Cas9-2. And let's just look at the second one. If you follow the arrow all the way out to the red line, that shows that it takes 2 hours to digest all of that DNA compared to 15 minutes with wild type. And then the last one, it takes 12 hours to go there. So we've slowed it down. And now the question is, will it still correct enough cells to work? And so we did the same assay where we plate the cells out. This is a new cord blood, new gene correction experiment. And fortunately, we found, we were very excited this day in the lab, that 46% of the cells were corrected, which is still high enough to cure the patient if they were transplanted. But the most important thing is now the indels over indels, that is the genes that were mutated by that, were only 10%. And we know that that is not enough to cause beta thalassemia. And then on top of that, we got 50% cell survival, and we can even increase that more. So these numbers suggest that we can do this safely and effectively in human cells. This is just the sequence of that, and in the top I have one cell from a colony where the sickle mutation is not corrected. If you look to see where the scissors are cutting between these two Gs, and so that's the PAM site, and that T just upstream of the G is the sickle mutation. So that is T, and what we want to do is correct it to an A. And if you look at the bottom one, you can see that we've corrected both copies of the T to an A, but most importantly, there are no non-specific changes around the cut site and the correction site. So 0 of the 96 colonies had non-specific modifications, where 5 of 96 colonies had these rearrangements before. So we felt like our modified recombinant Cas was what we wanted to use. Now, the question is, does the enzyme and the guide RNA and the Cas9, modified Cas9, do they ever cut off target? Do they cut in other places of the genome? So this is a specificity question. So we want high gene correction, low indels, on target indels, and then we want low off target cutting. As you know, nothing in biology is 100%, so there will be occasional off-target cuts. So we want to know where are those, and we want to know that they're not in any gene or gene regulatory element. And so we can determine where our complex will cut by doing a procedure called CircleSeq, and basically we just extract DNA from a sickle patient's blood, from one of these cord bloods, and we extract the DNA and we cut it up into small, into fragments of about 500 base pairs, and then we circularize them. So we make circles, and then we expose those circles to our complex that we have electroporated into cells. But now this is an in vitro experiment, and so then we put in the complex. It cuts where it's supposed to cut, and if it's going to cut somewhere else, it will. And then when it cuts the circles, it linearizes them, and then we put on linkers and sequence them. And this slide shows 3 sequences, off-target sequences, that we did cut, and I'll show you in a minute that it's cut very rarely. And so we determine the sequence of those, and those sites are not located in any known genes or regulatory elements. So we're very happy to see that. Here's off-target site 1, didn't cut at all. Off-target site with wild type, it cuts a little bit. With our modified, it cuts none. And then an off-target site 3 is cut by 5% of the DNAs are cut, of the alleles are cut, which is way, way too high. And with our modified recombinant, it's just 0.5%, which is low enough to meet the FDA's safety standards. And even those cut sites are not in any gene and don't disrupt anything. So, so that's one of the criteria that we'll have to meet. Here's the last one. We've done many of these in many different cord bloods from different individuals. And here, 60% of the cells were corrected. There were only 9% indels over indels where both genes were deleted, and 50% cell survival. So that's far above what we would even need to correct a patient safely. Now, gosh, I don't want to go too far, too much more, but we have to do another experiment, and that is we actually have to do a transplant. We can't transplant patients yet. So what we do is transplant the cells into a mouse that is immunocompromised. That is, they don't have an immune system, so the human cells can function in the mouse and replicate. So we transplant these NSG mice that don't have an immune system. The human stem cells can go to the bone marrow of those mice and replicate and make all different cell types. And we let that go on for 4 months. It's a long experiment. And after 4 months, then we collect those human cells from the mouse and ask if at what level have the sickle mutation been corrected. So this is just a fluorescent activated cell sorter data where we can purify the human cells. So after we collect the bone marrow from the mouse, we run it through the FACS sorter, and that, of course, we've bound fluorescent antibodies that distinguish the mouse cells from the human cells, and then the sorter sorts them into human cells, and then we look at myeloid cells versus erythroid cells. So that's our myeloid cells versus lymphoid cells. So that's in the right panel. The pink cells are CD19 positive on their surface. That means they're lymphocytes. And these green ones on the bottom right are positive for CD33. So those are human myeloid cells. And myeloid cells are, erythroid cells are included in that, in those cells. So it just points, these are two different lineages that have come from a single hematopoietic stem cell. So if they were corrected in both of these populations, we can surmise that they've been corrected in true stem cells. And in fact, they have, and this is a busy slide, but I'll just show you. If you look right in the middle, it says percent SLL correction, and you can see in the human CD34 positive cells, which is a marker for stem cells, we have very high correction efficiencies, far above 20 to 30% that's necessary. But the other important thing is percent S allele retention. And what that means is if the sickle gene is not corrected, we don't want it to be deleted, because that sickle hemoglobin, if it's in a low percentage of the cells, it will not cause the cells to sickle because it's competing with normal corrected hemoglobin. And that is, all we have to do is correct one of the two genes. So we will convert this individual from SS to AS. And remember, we know that's sufficient because if we do matched sibling bone marrow transplants, they're cured. So, but you need to maintain the S gene because sickle hemoglobin does bind and deliver oxygen well, and we need that oxygen delivery capacity. We just need to keep the level of hemoglobin S low enough that they can't form polymers, and so we can. Now, we've also done this in our mouse model of sickle cell disease. I don't have time to go through this. These are papers we published over the years. We humanized these mice by changing their alpha globin genes to human alpha globin and their beta globin genes to human gamma and beta. So these mice that you're looking at make only human hemoglobin. They make only human sickle hemoglobin, and we may have mice that make only normal human hemoglobin. They develop the full pathology of the disease. Here's a blood smear that shows classically sickled shaped cells. And if you examine their organs, they are filled with sickled cells, and they develop all of this pathology. They lose urine concentrating ability. They have strokes, they have everything that humans have. So they've turned out to be a great model, and we've distributed these all over the world. And so here is a slide of one of our sickle mice, and you can see these sickled cells, and then this is after we've done the gene correction. And these are spleens. The spleen on the left is normal control. The spleen on the right is from a sickle mouse because they're very anemic. They're very severe, and their spleens enlarge in order to try to make more red blood cells to correct the anemia. But of course, every cell that they make is still a sickle cell. But the one in the middle as after gene correction, so we can cure that animal model. Okay, I'm not going to talk about this. I might be able to handle it in the discussion, but in order to satisfy the FDA, we have to be able to do this all in a facility where every worker's gowned up. There are a huge number of regulatory issues in order to make it safe for patients. And so we have this facility at UAB. This is very near the bone marrow transplant unit or stem cell transplant unit. So the cord blood would come from the cord, would come into this room. All our gene correction would be done in this room. We would freeze 90% of the cells down, do all the tests that I described to meet the FDA criteria for safety and efficacy. And then once we've met all those criteria, then the cells can be thawed. The 90% of the cells can be thawed and transplanted into the patient to cure the patient. So this is just an acknowledgement of all the people involved. The people in my lab are listed at the top, and they're all phenomenal. Many of them were graduate students in my lab, and then were postdocs, and it's just a team of very dedicated scientists who want more than anything to cure sickle cell disease, not only in the United States, but we have collaborators in Uganda and in West Africa, in many places, in Africa and other countries, in order to do this process if the FDA approves it. So, all right, I probably talked longer than I should have, but I'm happy to answer questions.
Okay, we had a couple come in. Is there a treatment for this disease? That's the first question.
Yeah, so that right now, the only treatment is what is a drug called hydroxyurea, which patients take, and that can increase their fetal hemoglobin. There's no treatment that corrects the mutation, but long-term hydroxyurea treatment has side effects. All drug treatment for long periods of time have side effects. So this would be the first treatment of actually correcting the sickle gene in a sickle patient.
Okay. Here's another one. So BFU-E is a single cell, question mark?
Yeah, so it's BFUE is a progenitor of a red blood cell that's in the bone marrow. So the stem cell has committed itself to make a red blood cell, but that happens in a series of steps. And so those cells are rapidly dividing. And when we take them out and we plate them in this methylcellulose, which is like Jello, and give them all of the growth factors that allow them to divide and grow, then a single cell can grow into 100 cells or 1,000 cells. And then we can pick that and extract DNA so that we can do DNA sequence. And that's how we can count the fraction of cells that are corrected. So that's a BFUE. We all have BFUEs in our bone marrow right now, hundreds of thousands of them. And those are the cells that are just pumping out red blood cells. But we capture them in this Jello in order to analyze them.
Okay, here's another one. What challenges have you encountered with culturing hematopoietic stem cells? Also, have you encountered rejection of grafted cells or other challenges with getting cells to take the host?
Yeah, so that's a great question. We purify really hematopoietic stem and progenitor cells. They're not 100% pure hematopoietic stem cells. But in the cells that we isolate now, about 1 in 100 are true stem cells, but we want the other cells around to protect the stem cells. And so, and if we can get 20 to 30% correction in the stem cells and in progenitors, then we know that those stem cells can survive because they're going back into the same individual that they came from. So this is an autologous transplant. So we're taking cells from the cord, but those are the baby's cells, and then we want to transplant them into that same baby. So this is an autologous identical match, except that the sickle gene is corrected. Unfortunately, we cannot expand those stem cells. What we would love to do is do the gene correction, identify the corrected cells, and then expand them to a level that we could transplant only corrected hematopoietic stem cells. But that's impossible now. But we really don't have to do that in order to cure the patient.
When do you see this gene correction treatment to be approved by the FDA and be an option for patients?
Yeah, that is a great question, and I hope this meeting on March 2nd goes well, and I think it will. They may ask us to do some more experiments. Typically they do, and we would have to do that. For instance, if they ask us to do more individuals now, the same way that we've already done, those experiments take 4 months to do. And so it would take a year to get that. If they ask us for more data, it would take another year to get that. And then we would have to reapply to them to get approval. And then it takes some time in order to actually get the clinical trial done. So I would say it's 1 to 2 years. But that's actually fairly rapid. We want to be safe, but also we know that that over 1,000 babies are born in the United States a year with sickle cell disease, and tens of thousands in Africa. And the lifespan of a sickle patient in the United States right now, the average lifespan, because of excellent supportive care, is 40 years old, 42 years old. The problem with that is that there are more and more complications the older the patient gets. In Africa, the average lifespan is 5 years. I mean, we just have to be able to do this in Africa in a safe and efficient and cost-effective way. And that's another thing that we've focused on, being able to do this cost efficiently. That is, we're making a biochemical complex. We can synthesize, manufacture these 3 components: the guide RNA, the Cas9, and the single-stranded oligo, which is a correction template, and make that complex. So it shouldn't be that expensive, and that's a critical that's a critical consideration, because doing this in adults with the gene addition therapy now is over $1 million, and that obviously just can't be done worldwide. So,
what made you interested in sickle cell diseases?
Yeah, I guess I originally, well, there are 2 things, and one is that I knew patients with sickle cell disease, and I just saw how devastating it was for the patient and for the patient's families. And then secondly, on a scientific basis, I thought that in this particular disease, every person in the world who has sickle cell disease has the exact same mutation. That A is changed to a T. And so we could develop one therapy that could correct them all. There are so many other diseases involve several different genes can cause the same pathology. For instance, in beta thalassemia, there are 500 different mutations that can cause the disease. And so that means you'd have to do 500 different corrections in individual patients. This, we have one that works for all.
Okay, I think you just touched on it, but is this only for sickle cell or also for thalassemia?
Yeah, it's definitely for beta thalassemia. So it's the same idea. The only thing we change is the guide RNA. We just put the gene that's causing the thalassemia in that patient in the guide RNA, so that the guide RNA goes to migrates to that gene, and then the correction template is this new sequence that we want it to be. And so we can do that. I mean, those again are just synthesized. But we also think that this will be effective for diseases like severe combined immune deficiency, where kids are born and have no immune system. And those kids die within 3 months if they don't get a matched donor transplant. But then very few of them have matched sibling donors. And so, but many times that's a single base mutation in a single gene. And therefore we can make a guide RNA to that, excuse me, and a correction template to that.
Okay. Can you test a fetus for sickle cell disease prior to birth? If so,
that's a great question. And the answer is yes. And that is done very safely now. And we've done it at UAB in collaboration with our OBGYN and maternal and fetal medicine group. It turns out that in every mom, there are some fetal cells lyse and their DNA spills out into mom's blood. So it's a very small amount. The DNA is chopped up, and so it doesn't have any effect on the mom, doesn't have effect on the child. But if you can get 10 cc's of blood from mom, even as early as the first trimester, but works best in the second and third trimester. The vast majority of, you get the 10 mils of blood and spin it down and take the plasma off. So the cells you throw away. And the plasma has mostly mom DNA in it that is there because cells lyse, and we all have DNA in our plasma. But there's a small amount, maybe 1 to 3% of this DNA is from the baby. And it turns out now there is a company actually who has perfected a way to sequence that DNA and actually diagnose a child before birth. And that would really be great in this particular instance, because then we would know which cord blood to collect. Right now, we collect all cord bloods from mothers that are carriers. And only 1 in 40 of those are SS, a sickle patient. So we have to collect 40 to find the one. But with this other method, we can determine which mom is having an SS child.
Okay, here's another one. What made you interested in finding a cure for sickle cell disease? Was it curiosity or was there a firsthand experience?
Well, there's, I sort of answered this before. It's two things. It's knowing patients and families. I guess that's the social reason that I just saw so much pain and suffering, but the other reason is there's actually two scientific reasons. One is that every patient has the same mutation. Therefore, if we develop the correction of that one, we only need one approach. But the other one is that I've also always been interested in the mechanism of the switch from fetal to adult hemoglobin. And so that's really the way I started my scientific career, is to try to understand how that switch was accomplished. And we were thankfully very successful at that. And we defined the regulatory elements that are required for that switch to happen. And we identified regulatory sequences that are 50,000 base pairs away from those genes that are critical. And we actually, from our data, developed the model for globin gene switching from fetal to adult switching that still stands. So, and it's actually one of the bases for that is what the people are using to do the fetal globin reactivation clinical trials for sickle cell disease. But I guess in the end, even though we figured out a lot about how, and of course there are other groups that contribute a lot, but we, I think, first showed globin gene switching. We did it in a transgenic mouse where we could microject the human genes into a fertilized mouse egg, make a mouse, and show that it switched from human fetal to human adult hemoglobin. Then we could go back and make mutations we made thousands of mice where we made mutations to see if we could disrupt that switch, which is how we identified the regulatory elements. So that scientifically was very satisfying. But that's just kind of not enough for me. I want to have, we really want to do something absolutely for sure that can cure a patient in the end. So curiosity is important, and trying to relieve suffering is important as well.
Okay, two more. Do you think in the future you could do non-identical transplants? Like say someone donates their cord blood for someone else's baby?
Yeah, and cord blood transplantations have been done, and they're actually, those are called allogenic because they're from a different individual, and they can be typed so that they are they're as matched as they could be without being identical, and those transplants have been very successful. Unfortunately, that it's there are still problems with allogenic transplants, and but because you have to suppress the transplant from attacking the recipient cells. That's called graft versus host disease. And when the graft comes in, it obviously is even if it's fairly well matched, it's not identical. So it does recognize this recipient as foreign, and there are, and then the patient has to be immunosuppressed, and that can make them susceptible to viral infections. And but I will say that can work, and it should be pursued, but it's not as good as an auto transplant. So.
All right, last one. During surgery, do sickle cell patients who need a blood transfusion, is there any altercations involved with that?
They typically do have transfusions, and in fact, very many sickle patients get monthly transfusions, and so that's very, very common. And yes, they do get transfused before surgery, absolutely, because they don't want to put a patient under anesthesia that's making 100% hemoglobin S in their red cells. So they transfuse them so that they have, more than 30% normal red blood cells, so that and that prevents their other cells from sickling, because it gives them a lot more oxygen capacity. So the answer is yes, they're virtually all transfused before surgery.
Okay, no further questions.
Okay, well, thank you for your attention.
Yes, thank you very much, Dr. Townes. Thank you very much. We really enjoyed the talk. We'll see you again.
Okay, very good. Thank you for the invitation.
Not at all. Thank you.
Okay, goodbye.
Goodbye. Yes.